Alexandra Rubin, and I will be presenting the research I've been working on the past year on mycorrhizae quantification in sand dunes on Sapelo Island, Georgia. So the motivation for this research is to quantify how many infection rates that we see in the mycorrhizae in this sand dune in particular, because it has been subject to hurricanes in the past 5 years or so. So the methods and materials that were used: first we went out on site to the island and extracted the roots from the ground. Then the roots were put directly into a cooler and then in ethanol to preserve them. And then we followed the staining procedures that have been found in past literature reviews. The staining procedure uses KOH in a water bath, and then it goes into hydrogen peroxide and then HCl. Once this is done, we stained them in an ink vinegar solution instead of using Type-in blue, because we found in literature that that can be a carcinogen. So as you can see in the presentation, there's a study site. So there is a map here showing where in the world Sapelo Island is. It is off the coast of the Atlantic Ocean in Georgia, and it is a small island. Once again, this spot was chosen because it has been subject to hurricanes in the past. So in the experiment, we looked at 2 different types of plants. The first plant we looked at are sea oats, and the second are spartina. So you can see images of these 2 different types of plants here. So the results from this research are found on the right of the poster, and you can see in figure 5 that the average proportion of vesicles across each plot type were found. Then in figure 6 you can see the average arbuscules, and in figure 7 the average hyphae. So overall there was the biggest proportions found in vesicles and in hyphae, and so this is because we have found through literature reviews that arbuscules have a shorter term of life, and so once they're there, they go away after a short period. About a week or so is their lifetime, and then are typically replaced by vesicles. So in conclusion, there is a high infection rate of mycorrhizae found in the dune, and so it's still relatively healthy even though it's been impacted by hurricanes. Thank you very much.